During this presentation, I will continue my discussions on the methods used for the isolation of cells from fat, with a particular emphasis on the variability that exists in the literature and different laboratories performing this isolation. And then I'll finish with some recommendations on how we can standardize these different methods for isolation of these cells. Over the numerous decades since the original publication by Martin Rodbell of collagenase digestion of fat for the isolation of cells, there have been over 13,000 publications that utilize the term adipose cell and isolation. And many of these publications do describe variations in methods for the isolation of cells from fat using enzymatic as well as non-enzymatic methods. Now, what I've been doing is reviewing all of these papers, and I've created a database, and here you see the headers for this database. In the database in my review, what I've been evaluating is all of the individual variabilities for the isolation and subsequent characterization of these cells. And in this database, I've been focusing on just this primary methodology for the isolation of the cells. I'm also doing a separate study where I'm evaluating variations in the culturing of these cells, the characterization of the cells, and ways of doing specific subfractionation and passage of the cells for research as well as clinical use. So I'm going to take you through some of the findings that and conclusions that I've come up with on the variability of these isolations. Now, I talked about this in a previous slide, but I'll show this slide again. This is the variability in the terminology used to describe the original pellet that results from what I call the Rodbell method of 1964. Collagenase digestion of fat, and then a centrifugation resulting in a pellet, which Marty Rodbell originally called the stromal vascular cell pellet. Since that time, now through 2025, there have been numerous investigations where this pellet of cells, the stromal vascular fraction, has been called many different things. I give you the quote from Shakespeare on a rose is a rose. 64 Rodbell again, stromal vascular cells, and I've reviewed all of these different methods previously, but the terminology is incredibly variable. Sedimented cells, capillary stromal cells, vascular stromal cells, microvascular endothelial cells, and it just goes on and on. The process lipoaspirate cells, stromal cell pre-adipocytes, stem cells. What it shows is there's incredible variability in how we characterize this cell population. Now, what I want to talk about now is the process variability, how different investigators, different groups use different methods for the isolation of what we'll call the primary cell isolate, which is going to be called, and I've talked about this before, either the stromal vascular fraction, the stromal cell fraction, or the microvascular fragment fraction following either enzymatic or non-enzymatic digestion. Here are all the variabilities that I have found in the technique for the isolation of these cells. It starts with species variability, human versus non-human sources of fat, variability in anatomic sources within individual species, and then you see all of these variabilities, how we store the tissue, mince the tissue, the enzymes we utilize, and the concentration of these enzymes, the digestion temperature, agitation, time, and enzyme filtering, the Bjorntorp method that was incorporated into this isolation, and how different cell populations can be filtered. centrifugation speed and centrifugation time, put those together and you end up with a G-force. We should not be publishing using RPM. We should publish utilizing a G-force. Washes, subsequent filtering after that, resuspension, and then many times these cells are used immediately or they are put into culture. Here are all the variabilities that exist in this procedure. And I've taken this one step further in this particular slide to show you a little bit more of a breakdown in these variations. I'll go through this quickly. You can pause in the presentation later and look at each of these individual variables. And I'm also going to be giving subsequent presentations on some of the specific variables you see here. Most notably, one of my next presentations is on tissue dissociation enzymes. But again, to give you an idea of some of the variations that I've found, 1964, the origins of the technique, to 2025, the fat has been isolated from numerous species, and within these species from numerous sites. That creates variability in the type of fat that is obtained. The mincing process, it really ranges from no mincing at all to liposuction as a form of mincing, homogenization, sonication, garlic presses. It's a huge variability. Enzymes, again, I'll talk about this more, but crude collagenases, crude enzymes versus purified enzymes, and then other proteases that can be added. The vessels that are used, the agitation, incredibly important. And the variation I've seen has been from no agitation. The fat is simply put in with enzyme and incubated at 37 degrees with no agitation, or with very significant agitation with the addition of certain materials such as marbles and beads in order to try to break this tissue up as quickly as possible. Time differences, 4 minutes to 24 hours in enzyme. Filters, numerous filters have been utilized over the years. Gauze, cheesecloth, and then different micron size filters. Centrifugation from 1G, which means just gravity separation of the pelleted cells, up to 1,000 times G for a short or relatively long period of time. Purifications, a variety of different methods have been reported, including fluorescence activated cell sorting, Percol, Ficol, Nicodens, a variety of different medias. Secondary filtering, and then finally the final product has been called many different things. You see on the right bottom side of the slide, stromal vascular fraction to process lipoaspirate and everything's in between. What I have tried to do is present a concept that we standardize what cell population we get from fat that is either enzymatically or non-enzymatically isolated. Enzymatically, we can either get the microvascular fragments, capturing those cells on a filter, a stromal vascular fraction by using no or large filters, or the stromal cell fraction, utilizing filters to remove the endothelial cells, so you get a relatively endothelial cell free. One last one in this variation I want to point out is once you have this final product, microvascular fragment, stromal cells, stromal vascular fraction, and you send it off to the fluorescence laboratory, the cell sorting laboratory for characterization, they will often utilize a filter before putting the cell population into a fax scanner or a fax sorter. And those filters can be as small as 20 microns. That will effectively remove a significant portion of the endothelial cell component of the cell isolate that you have. So you see an incredible amount of variation in the methods, and it is very, very rare that a publication, a method, is presented where all of these variations are provided in detail, which makes the comparison of one group's isolation technique to another group's isolation technique extremely difficult. I've taken a look at the literature and evaluated the net yield of cells per gram of fat. You see this is times 10 to the 6 cells per gram of fat, so million cells as here. And then you see from Rodbell, and Rodbell of interest, the number of cells that he calculated in the vascular stromal fraction was over 10 million cells per gram of fat. He did this based upon a direct DNA analysis. You see, over a period of time, this has been ordered from 1964 through 2024, you see a relatively tight distribution of cell yield around 1 million cells per gram of fat. I also give you a bar here, which is what I consider the literature bar, the potential human SVF cells per gram of fat. And this is based on in situ cell quantitation, where investigators have taken a piece of fat, have sectioned it, and then determined on a volume basis the number of cells per volume of fat that has been evaluated, and you get around 3.5 million cells per gram of fat is the potential yield of SVF from a gram of fat. That means if you are completely efficient, 100% efficient in isolating cells from fat, you will get around 3.5 million cells. Plus or minus what I would say is about 4 million cells. There's quite a variation. This is relatively tight because it's only based on 2 publications that are out there, but there is the possibility of some of this in situ fat containing more than 5 million cells per gram of fat based upon inflammatory cells and other cells that may be present. This is the published relative distribution of different types of cells in fat once you've done an enzymatic isolation of SVF, SC, or MVF. Talked about this briefly before. This characterization just of endothelial cells alone goes from 0% endothelial cells in the final SVF fraction up to 100% of the cells are endothelial cells. Reality is somewhere in between in all of these cell populations as far as what the factual number of individual cells on a percentage basis of the total cells that have been isolated. And there is a fair amount of controversy as far as like presence of parasites. As I've talked about before, there are very, very few parasites in the microcirculation of fat. So investigators that find 15%, that's about the high number I've found, 15% parasites per total population of FVF cells. This is probably due to errors on the markers that are used to identify these parasites. We just don't see them in situ inside the fat. So there's your variation that goes on there. I do want to take a moment to talk about this release of cells using enzymatic or non-enzymatic means. And do you get all of the cells in fat removed from the fatty tissue using non-enzymatic methods? And the answer simply to that is no. Non-enzymatic dissociation of fat using liposuction or passing the fat through a homogenizer or very tight orifices in order to break the tissue up, you will not release the vascular fraction. If you call a non-enzymatic dissociation of cells, that cell population, the stromal vascular fraction, I believe that is incorrect nomenclature. It is a stromal cell population, but as Bjorn Torp pointed out in the 1970s, putting these filters in or not using enzymes will not release the endothelial cells, or the endothelial cells will be removed, his filtering methods, during the processing. Only enzymatic dissociation will release the endothelial cells from fat. So enzymatic versus non-enzymatic tissue dissociation. Enzymatic dissociation is required to release the vascular, the endothelial cell fraction. Non-enzymatic methods do not yield stromal vascular fraction. They yield the adipose stromal cell fraction. Now, again, I will present a unified terminology for cells isolated from adipose tissue. The original Rodbell method, and this is the top method, enzymatic digestion using collagenase and protease. This could be a crude collagenase that contains both class 1, class 2 collagenase and protease, or it could be purified, ultra-purified, or recombinant collagenase added to purified proteases. Enzymatic digestion over 20 minutes and using either no filter, or I've set the discrimination point as a 200 micron filter. If you use this filtration, no filtration, or this filtering method, it results in an adipose stromal vascular fraction cell population, and give Rodbell the primary reference to the method that is used. Bjorntorp, he puts filters in, and these filters are less than 200 microns. For that reason, Bjorntorp stated it, and I agree with this, it is an adipose stromal cell population, not the SVF population. Bjorntorp referenced Rodbell in his paper and said it's the Rodbell digestion method, but the Bjorntorp filtration method to result in the adipose stromal cells. Wagner taught us how to utilize a filter to capture the microvascular fragments. It is what I'd call a less rigorous enzymatic dissociation over a shorter period of time. He captured the fragments and he called these the adipose microvascular fragments. Roger called these the capillary endothelial cells. That's the Wagner method. Wagner also referenced Rodbell for the digestion process, but it's the Wagner filtration process to capture these cells. And then finally, non-enzymatic tissue disruption. This is something that Alexis Carrel provided methodologies for dating back to the 1920s. If you utilize the Carrel method to isolate cells non-enzymatically, those cells should be described as the adipose stromal cell population. They are not the SVF population. So this is an attempt to head towards a standardization of all of these methods. I want to point out, as far as methodology, some of the things that need to be avoided in the presentation of methods. One, don't simply use the term adipose cells were isolated using enzymes. And in my review, I've seen some investigators say that that's their method. They took adipose tissue and they used enzymes to isolate adipose cells. This doesn't provide any sort of information on the method that is being used. I've seen SVF cells were isolated according to standard methods. It'd be nice if we had a standard method, but without it, this is an incorrect methodologic statement. I've seen SVF were isolated according to X, citing a previous paper with the statement with minor modifications. I don't find this acceptable also. I think, the SVF, if they're isolated, the method that more than likely is used is the Rodbell method. And then you need to state exactly what modifications have been used. Bjorntorp did it, Wagner did it. All the original investigators utilized the Rodbell publication and then gave their very specific modifications. I don't think it's appropriate to say just with minor modifications. And then I've seen SVF isolated according to X, where if you go to reference X, it will cite reference Y. And then if you go to reference Y, it will reference a paper by a different set of authors, reference Z. I've carried this out, and the largest string has been 6 different referencing, citing a reference, citing a reference, and the original reference provides very little information. It is unknown what true methods were being used to isolate these cells. So this is my belief as far as standardization of adipose cell isolation methods. When you publish, you should provide all of this information, starting with the species, source of the fat, how it's isolated, all the different variables I've already talked about should be provided in detail. When you give centrifugation, don't give just RPM. It needs to be G-force. And you can go back and take a look, and this is what I hope this field will move towards as far as standardization of the methods. And then the cell populations that are released are either the microvascular fragments, the stromal vascular fraction, or the stromal cell population. And then we have something else that looms ahead. This is the cloud of FDA, that once we have these cells isolated, regardless of what we call them, and I'm pushing towards these 3 terms, SVF, MVF, and SC, the FDA, before we take this into clinical application, they are going to want to know identity, strength, quality, and purity of this cell population. It then becomes a process where we have to develop standardized methods for the characterization of the cell products that are isolated from adipose tissue. That initial characterization can give us CD markers, a variety of different characteristics. We then have to determine potency of this cell population, and then based upon the potency, we've got identity and strength. Based upon that, then we can begin to talk about dosing of this cell population. So we still have a long way to go to standardize this. So thank you for your attention. I hope you enjoyed this segment. Again, if you have any comments, my email address is given below, cvregen@gmail.com, and then you can find this publication and all these publications, or presentations, on my website at cvregen.com. Hope to hear from you. Thank you.